Very good afternoon, students. Today, let us move on to the next topic, that is citric acid cycle, or even it is also called as TCA cycle. So, why this TCA has been referred as TCA cycle or the citric acid cycle? So, the citric acid cycle is one of the metabolic hub of the cell. It is the final common pathway for the oxidation of fuel molecules such as amino acids, fatty acids, and even carbohydrates. As you all know that in case of eukaryotes, the reactions of these citric acid cycle which is going to take place inside the mitochondria, where we have observed the glycolysis or the glycolytic enzymatic reactions which has been occurred or which we have seen in the cytosol. So this citric acid cycle is also called as tricarboxylic acid cycle because tricarboxyl compound is going to be formed here, that is called as citrate molecule. That's why we are going to call it as citric acid cycle or the tricarboxylic acid cycle. It is a series of reactions which are going to happen in the mitochondria, as just now I said, which brings about the catabolism of acetyl residues to carbon dioxide and water in case of aerobic condition. So this TCA cycle also happening aerobically, aerobic process it is, where the hydrogen equivalents upon oxidation, which leads to the release of most of the free energy, which is going to be captured as ATP molecule, that is in the form of ATP, which is the most available form of energy for the for all the tissues. So these are going to act as the tissue fuels. So the acetyl residues which are in the form of acetyl CoA, this is an ester of coenzyme A which contains the vitamin pantothenic acid. So the major function of this citric acid cycle is to act as the final common pathway for the oxidation of almost carbohydrates, lipids and protein molecules. So this is because glucose as well as fatty acids and many amino acids are all metabolized to acetyl CoA or the intermediates of TCA cycle. And even along with these informations, so this TCA is also amphibolic in nature. Why is it so? Because this is going to act both catabolically and as well as anabolically. That is amphibolic in nature. Both anabolism and catabolism is going to happen. So the next question here it arises is, what are the sources of acetyl CoA molecule? So as I said you in the previous slide just now, so the coenzyme A, which is abbreviated as CoA, which is mainly composed of beta mercaptoethylamine pantothenic acid, that is a vitamin, and adenosine 3-phosphate and 5-diphosphate, right? So we have seen so far in all other metabolic pathways, that is glycogen, we have not seen this glycogenesis or the glycogenolysis. In the future classes we can see. So where the particular acetyl CoA molecules which are going to be produced from where and all. So the glycogen is also acting as a precursor. Through glycogenolysis process, you are going to break down this glycogen to form glucose molecule. So this glucose is entering into the glycolysis. This is going to result in the formation of pyruvate, that is catabolism is happening. Glucose is degrading into pyruvate. Then this pyruvate which results in the formation of acetyl CoA. So this pyruvate will be the source for the acetyl-CoA. Initially, glycogen was the source. From the glycogen, glucose is going to be produced from the glucose pyruvate molecule, and this pyruvate indirectly it is going to act as a source for the acetyl-CoA. Along with this pyruvate, it also includes fatty acids and amino acids, as I said you earlier. So these fatty acids molecules are also involved in the synthesis of acetyl-CoA, fatty acid metabolism, and amino acids degradation. It also results in the production of acetyl-CoA molecule. Some amino acids will give you acetyl-CoA. It plays a major role, this particular pathway plays a major role in the transamination reactions, gluconeogenesis reactions, deaminations reactions, and lipogenesis, where several of these processes are going to be carried out in many tissues, but the liver is the only tissue in which all occurs to a significant extent. So these reactions of the citric acid cycle liberates reducing equivalents and carbon dioxide molecule, where the reactions of this particular TCA cycle we will see now. What are all the following steps which we are going to come across in this TCA? First and foremost will be the formation of acetyl-CoA from the pyruvate, where this pyruvate has been generated through glycolysis process. So after the glycolysis, once it is formed in the cytosol, now it is undergoing pyruvate dehydrogenation reaction catalyzed by pyruvate dehydrogenase enzyme, which requires cofactor NAD plus, and it involves coenzyme A, and carbon dioxide is going to be released. It is undergoing oxidative decarboxylation process. So this PDH, which is not a part of TCA but important for the supply of for the supply of acetyl-CoA molecule. So which plays a very important role in this pyruvate dehydrogenase enzyme or pyruvate dehydrogenase, which is acting as a multi-enzyme complex. So once after the formation of this acetyl-CoA molecule, now the first enzymatic reaction of TCA starts. So here you can see here the condensation of acetyl-CoA with the oxaloacetate to form citrate. That is the first enzymatic reaction which is going to happen in the mitochondrial matrix. Once after the formation of acetyl-CoA, it is entering into the mitochondrial matrix, and there it is going to be acted upon by the enzyme citrate synthase by condensing with oxaloacetate. Acetyl-CoA is going to condense with oxaloacetate to form citrate, so catalyzed by enzyme citrate synthase. So coenzyme A is going to be removed. You can make out here. So acetyl-CoA is condensing with oxaloacetate. You can see the structure of oxaloacetate, CO minus, C double bond O, CH2CO minus. So coenzyme A is going to be removed. You can see here from the acetyl-CoA, acetyl group is going and attaching to your oxaloacetate to form citrate. CO minus, CH2COH, CO minus, CH2CO minus. This is the structure of citrate. Coming for the next enzymatic reaction, conversion of this citrate, which is going to be resulting in the formation of isocitrate via the formation of intermediate that is called as cis-aconitate. Cis-aconitate is going to be formed in between the citrate and isocitrate molecule. That is the intermediate which you can come across. The citrate is getting converted to isocitrate by the enzyme aconitase, which is undergoing first hydration reaction. right? The enzyme is aconitate hydratase. So you are adding water molecule to the citrate molecule. So hydration reaction is happening, catalyzed by aconitase, which contains Fe2 plus ions. So here you are adding water molecule to form the intermediate, that is cis aconitate. Then dehydration of this particular one second, sorry. So first you are going to see dehydration reaction here. First you are removing the water molecule. So you can make out here from the citrate. So you are removing the water molecule. Then you are going to hydrate, right? Rehydration. So this reaction takes place in two steps. That is, first is dehydration to form cis aconitate, and then further rehydration to form isocitrate. So you can see here. So the citrate molecule CO minus CH2 CHCO minus CHOH CO minus, right? So for this you are removing water molecule from this citrate to form cis aconitate. This cis aconitate, which is going to be getting rehydrated further to form isocitrate. So you are adding water molecule that is again attached to that CH, to that place OH. Again here one more H. So you can see here CH2 CO minus CH2 COH CO minus CH2 CO minus. So you can see the formation of isocitrate. Rehydration reaction has happened to form isocitrate. Coming for the third enzymatic reaction, that is dehydrogenation of isocitrate to oxaloacetate. So first the dehydrogenation reaction of isocitrate is happening to form oxalosuccinate. Again, this oxalosuccinate is going to act as an intermediate. So here you can make out it is undergoing first dehydrogenation reaction in the presence of enzyme isocitrate dehydrogenase to form oxalosuccinate. So the linked oxidation of this particular isocitrate, which is going to proceed almost completely with the involvement of this cofactor NAD+. So this is an NAD+ dependent enzyme, that is isocitrate dehydrogenase. So you can see here in this slide, isocitrate is undergoing dehydrogenation reaction, where you can come across isocitrate dehydrogenase enzyme to form an intermediate oxalosuccinate. In between isocitrate to alpha ketoglutarate, you will have one intermediate that is called as oxalosuccinate. NAD+ is the cofactor here, or the coenzyme which has been utilized by this enzyme isocitrate dehydrogenase. So you can make out in the oxalosuccinate reaction, so CO minus CH2 CHCO minus C double bond O CO minus, right? So you can see here in this reaction how the particular dehydrogenation reaction is happening, right? Removal of, here you can make out the hydrogen which has been removed from this place and as well as from this place, right? So that is the 4th carbon atom, right? Coming for the next, decarboxylation of oxalosuccinate. Now after dehydrogenation, it is undergoing decarboxylation to form alpha ketoglutarate. So in this you can see here, so this particular step follows decarboxylation, as I told you. So that is nothing but removal of carbon dioxide molecule. So this is also catalyzed by the same enzyme, that is isocitrate dehydrogenase. That was the intermediate which is so formed. Oxalosuccinate is the intermediate which is formed from isocitrate through dehydrogenation reaction. Now decarboxylation reaction is happening from this intermediate molecule, that is oxalosuccinate. Carbon dioxide molecule is going to be liberated, right? So this particular enzyme also requires the metal ion Mn2 plus or Mg2 plus, which is an important component to carry out this decarboxylation mechanism or the decarboxylation process. So you can see in this oxalosuccinate conversion to alpha ketoglutarate removal of carbon dioxide molecule, you can see here carbon dioxide is going to be removed, right? Okay, decarboxylation has happened after dehydrogenation. Coming for the fifth step of this TCA. So here decarboxylation of this alpha ketoglutarate again is undergoing to form succinyl-CoA. So here for this decarboxylation of alpha ketoglutarate requires the enzyme alpha ketoglutarate dehydrogenase, right? Again the dehydrogenation reaction is happening, that is oxidative decarboxylation is happening. You can see here this particular reaction is carried out by this enzyme, that is alpha ketoglutarate dehydrogenase enzyme or complex, which requires cofactors such as thiamine pyrophosphate, lipoate, NAD plus, FAD coenzyme, same like PDC complex, that is pyruvate dehydrogenase enzyme complex. It also requires 5 coenzymes or cofactors, that is TPP, lipoic acid, NAD plus, FAD and coenzyme. So this particular reaction resulting in the formation of succinyl-CoA, which is an high energy thioester molecule. This is a high energy thioester molecule, and where you can see the synthesis of NADH, right? The NADH will be synthesized here. So here one more thing you can see here, this arsenic molecule, that is arsenate or the arsenic, which is a metal ion, heavy metal ion, which is going to act as an inhibitor, which inhibits this reaction, causing the substrate alpha ketoglutarate to accumulate. So it will go and bind to this particular enzyme substrate site, and it will block the mechanism of this enzymatic reaction, thereby alpha ketoglutarate is getting accumulated and it stops the TCA cycle, right? Further proceeding of TCA cycle or the tricarboxylic acid or the citric acid cycle. Coming for the next reaction, where you can see here conversion of this succinyl-CoA to succinate. What kind of reaction is happening here? So here you can see this succinyl-CoA is getting converted to succinate by the enzyme succinate thiokinase. Again kinase reaction is happening. So it is also called as succinyl-CoA synthetase. Synthetase involves ATP molecule or GTP. So here directly ATP is not going to be involved in this enzymatic reaction. This enzyme is very specific for, and it is going to utilize GTP molecule. It is going to synthesize GTP, right? So here you can see here high energy phosphate, where ATP is going to be synthesized at the substrate level phosphorylation reaction, which we have seen in case of your glycolysis. So can you tell me which was the substrate level phosphorylation in case of glycolysis? So what we have discussed, those three reactions, right? The glycerolidate 3-phosphate to 1,3-bisphosphoglycerate synthesis of ATP, that is considered as a substrate level phosphorylation. Another one enzymatic reaction is formation of involvement of phosphoglycerate kinase, that is formation of 3-phosphoglycerate, that is from 1,3-bisphosphoglycerate to 3-phosphoglycerate. Their synthesis of ATP, that is also considered as substrate level phosphorylation. And one more pathway is last, that is phosphoenol pyruvate to pyruvate. You can see again kinase reaction, they are also synthesis of ATP. Those three are substrate level phosphorylation reaction in glycolysis. So now we will see this ATP is not going to be synthesized in this as a substrate level phosphorylation. Here oxidative decarboxylation of this particular alpha ketoglutarate is going to be happening in that. So the reaction, in this reaction, it requires GDP molecule or the ADP, which is getting converted to GTP or ITP in the presence of inorganic phosphate, which then it is getting converted to ADP to ATP. That is from ADP to ATP. Synthesis of ATP, that is nothing but indirectly synthesis of GTP is going, synthesis of ATP is happening indirectly. So this reaction requires GDP molecule which is getting converted to GTP. So you can see succinyl-CoA thiokinase reaction which has happened here. Succinate is going to be formed and coenzyme A is going to be removed. So coming for the next reaction, dehydrogenation of succinate to fumarate. So here the succinate is further metabolized by undergoing dehydrogenation, which is catalyzed by succinate dehydrogenase. So it is the only dehydrogenation reaction in the citric acid cycle which involves the direct transfer of hydrogen from the substrate molecule to a flavoprotein without the participation of the NAD+. So without the participation of NAD+, you can see this is the only dehydrogenation reaction which is happening in the citric acid cycle, which involves the direct transfer of hydrogen from the substrate molecule. You can see here this reaction requires FAD and iron sulfur protein molecules, right? So this reaction which involves the enzyme succinate dehydrogenase, which is specifically requiring this FAD as a cofactor, FAD plus instead of NAD plus. So here you can see the formation of fumarate molecule. You can see the trans form of fumarate, not the cis form. So CH double bond, CHCO minus, 2 carboxyl group. You can see in this structure fumarate. Coming for the next reaction, that is the 8th reaction of TCA cycle, where you can see here addition of water molecule in order to break the double bond which is formed in between fumarate to give malate. So this reaction is catalyzed by the enzyme fumarase. It is also called as fumarate hydratase. Hydration reaction is happening. We are adding water molecule to break this double bond to form your malate. So in addition to being specific for the L isomer of this malate, fumarase also catalyzes the addition of the elements of water to the double bond of this fumarate in the trans configuration. You can see here the trans configuration of fumarate. So water molecule is added, this double bond is broken down. You can see, you can make out in the second carbon atom CHOH, right? So in the structure of malate. Coming for the next enzymatic reaction, that is dehydrogenation again. Dehydrogenation of malate to form oxaloacetate. So this malate which is formed is getting converted to oxaloacetate by dehydrogenation catalyzed by the enzyme malate dehydrogenase. This reaction requiring again NAD+. So this NAD+ molecule which is required by this enzyme malate dehydrogenase to form oxaloacetate, right? So the formation of oxaloacetate you can observe and synthesis of NADH. So oxaloacetate molecule which is so formed, it is going to enter into the gluconeogenesis pathway, right? The citric acid cycle end product, that is oxaloacetate, is now formed, is entering into the gluconeogenesis. So one turn around the citric acid cycle is going to be completed. An acetyl group containing 2 carbon atom is going to be fed into the cycle by combining it with oxaloacetate. At the end of the cycle, a molecule of oxaloacetate which is going to be generated, right? Oxaloacetate was generated. So the enzymes of this citric acid cycle, except for the alpha ketoglutarate and succinate dehydrogenase, are also found outside the mitochondria. So as a result of this oxidations catalyzed by dehydrogenase enzymes of the citric acid cycle, 3 molecules of NADH. So you have observed while explaining this TCA cycle enzymatic reactions, 3 molecules of NADH and 1 molecule of FADH2 are going to be synthesized or produced for each molecule of acetyl-CoA catabolized in 1 revolution of the cycle. So these reducing equivalents are going to be further transported to the respiratory chain in the inner mitochondrial membrane. So you can see in the respiratory chain how this ATP production which is going to happen. This involves the complexes, right? NAD plus coenzyme A, cytochrome B, cytochrome C1. You can see here in this slide, and you can understand this. So how it is going to happen? So during passage along the respiratory chain, reducing equivalents from each NADH generates 3 high energy phosphate bonds by the esterification of ADP to ATP in the process of oxidative phosphorylation. So however, this FADH2 molecule which is produced only 2 high energy phosphate bonds because it transfers its reducing power to coenzyme Q by passing the first site for the oxidative phosphorylation in the respiratory chain. So further high energy phosphate is going to be further generated at the level of cycle itself. So you can see at substrate level during the conversion of succinyl CoA to succinate, right? During the conversion of succinyl CoA to succinate, thus you can observe 12 molecules of ATP are going to be generated for each turn of the cycle. So you can make out in this slide how this energetics of the cycle you can see here. So how many molecules of NADH is going to be produced and how many ATPs are going to be formed from this, right? So the conversion of first is isocitrate dehydrogenase, you can see respiratory chain, oxidation of NADH and alpha ketoglutarate dehydrogenase, there also you can see synthesis of NADH, two molecules of so you can come across 3 molecules of ATP. So this is as per according to the later before and all you might have seen in the books. Now in advanced they are saying 1 NADH synthesis will release 2.5 ATP, according to that 5 ATP. So in the first and second enzymatic reaction, in this I am showing the 3 ATPs in the first and second 3 ATPs, and you can see here in the conversion of the succinate thiokinase, you have seen phosphorylation reaction at the substrate level which has happened, involvement of GTP molecule, that is synthesis of GTP, involvement of GDP, 1 molecule of GTP is synthesized. Here in the next you can see succinate dehydrogenase, there also 1 FADH2 which will release you synthesize 2 ATP molecules. In the last malate dehydrogenase enzymatic reaction, you saw 1 molecule of NADH synthesized. So 3 ATPs altogether, 3 plus 3 6, 7, 8, 9, 10, 11, 12. 12 ATPs are going to be synthesized, right? So this is the energetics of TCA. So you can see per 1 turn of TCA, 1 molecule of oxidation of acetyl CoA results in synthesis of 12 molecules of ATP. Net ATP is 12. Thank you. We will see in the next class other amphibolism of this, amphibolic nature of this TCA, as well as pyruvate dehydrogenase multi-enzyme complex mechanism and other enzymatic mechanisms in detail and the regulations of this TCA cycle. Thank you.